As you all know that DNA was first termed as nuclein. It was first termed as nuclein by the scientist Frederick Mischer. And we know that DNA is an acidic substance and it is negatively charged. It is negatively charged. Okay? DNA is a polymer, and it is a polymer of the repeating units, that is deoxyribonucleotides. Deoxyribonucleotides. And what are they? Let's discuss. Let's change the color, make it more colorful. DATP, DCTP, DGTP, and DTTP. These are the nucleotides of DNA. Deoxyadenosine triphosphate, deoxycytidine triphosphate, deoxyguanosine triphosphate, and deoxythymidine triphosphate. These are the nucleotides of DNA. So let's understand what is the basic structure of a nucleotide. A nucleotide contains three parts. First, a pentose sugar, and in case of DNA, this pentose sugar is a deoxyribose sugar. Second, it contains a nitrogenous base, and third, it contains a phosphate group. So these are the three constituents of the nucleotide of DNA. Nitrogenous bases further are of two types, that is purines and pyrimidines. Purines are of three types: adenine, guanine, uracil. Uracil is present in RNA, and adenine and guanine are present in DNA. Pyrimidines are of two types: cytosine, thymine. Okay, so let's understand the diagram of the nucleotide of DNA. In the center, you can see a deoxyribose sugar. This is in the center, it is a deoxyribose sugar. Now, why it is called as deoxy? Because you can see it. Let me number the carbons. This is 1' carbon, 2' carbon, 3' carbon, 4' carbon, and 5' carbon. 5' carbon of the sugar is present outside the ring. So, at the 2' position, beta, there is only hydrogen, not the OH group. That is why it is called as deoxyribose sugar. At the 1' carbon, of the sugar, the nitrogenous base is associated, and at the 5' carbon of the sugar, the phosphate group is associated. So, what you can say in this condition, that we can also use a term that is nucleoside. Nucleoside is equal to sugar plus nitrogenous base. As soon as at the 1' carbon of the sugar, the nitrogenous base get attached, it becomes nucleoside. And as soon as phosphate attaches at the 5' carbon, it becomes nucleotide. And in the case of DNA, the most appropriate word would be deoxynucleotide. Okay, so this is the basic structure. Now understand the bonds present inside one single nucleotide.
కొలంబస్ వెంచర్ కాలనీస్ నుంచి ఫోన్.
హోలీ షిప్, వాళ్ళిచ్చే ఫండ్స్ తో అమెరికాని చూస్తూ తిరుగుతున్నారు అనుకుంటారు. హలో యో హైనస్, లేదంటే ఇండియాలో చెప్పినట్టు నమస్తే అని చెప్పాలా? ఫోటోస్ ఫోటోస్ రైట్ సార్, ఖచ్చితంగా.
లమాస్ ని తీసేయండి తీసేయండి రా.
ఒక్క సెకండ్లో సార్ పంపించేశాను సార్.
మ్యాజిక్ ఎరేజర్ తో అవసరం లేని డీటెయిల్స్ ని ఫోటోలోంచి తీసేయొచ్చు. క్యాన్వా హృదయం నుండి రూపకల్పనకు.
The bond between the sugar and nitrogenous base is N-glycosidic bond. Is N-glycosidic bond, and the bond present between sugar and the phosphate group is bond between the sugar and the phosphate group is called as the phosphoester linkage. Phosphoester linkage. It's phosphoester, not phosphodiester. Okay, so within one single nucleotide you have two types of bond, that is N-glycosidic bond within single nucleotide we have N-glycosidic bond and phosphoester bond. Okay, between two nucleotides we have phosphodiester bond, and we'll do many questions associated with this topic in the MCQ section. So this is the diagram of NCERT, as you all must know. So let's understand what happens when nucleotide attaches and forms a polynucleotide chain. So beta, this is the sugar. As you can see, this is the sugar. Okay, this is the 1' carbon of the sugar, and this is the 5' carbon of the sugar. At the 5' carbon, phosphate group, 1' and the nitrogenous base. This is the N-glycosidic linkage, and this is the phosphoester linkage. Okay, N-glycosidic linkage and the phosphoester linkage. Now comes the phosphodiester bond between two nucleotides. This is one single nucleotide and this is the other nucleotide. This is the other nucleotide, and between two nucleotides we have phosphodiester bond. Phosphodiester bond. Okay, so many question comes regarding the calculation of number of phosphodiester bonds in a particular DNA. So let's deduce a general formula for calculating the number of phosphodiester bonds. As we all know, beta, we can have 4 types of condition in the DNA. That means the DNA can be double-stranded linear. It can be double-stranded linear. The DNA can be single-stranded linear, or it can be double-stranded circular, or it can be single-stranded circular. We can have these 4 conditions in the DNA: double-stranded linear, single-stranded linear, double-stranded circular, and single-stranded circular. So now we want to
The fashion and jewelry space is booming, all set to become a $1 trillion economy by 2025. And there
calculate the number of phosphodiester bonds. To calculate the number of phosphodiester bonds, let's have a general formula. In this condition, beta, double-stranded circular condition, we will calculate the number of phosphodiester bond by the formula. Number of phosphodiester bonds will be equal to number of nucleotides minus 2. Number of nucleotides minus 2. In case of this condition, when the DNA is single-stranded linear, then the number of nucleotides will be equal to, number of phosphodiester bonds will be equal to number of nucleotides minus 1. In case of either double-stranded circular DNA or single-stranded circular DNA, in both the cases, number of phosphodiester bonds will be equal to number of nucleotides. Okay? So, in the DNA having the double-stranded linear condition, number of phosphodiester bonds is equal to number of nucleotides minus 2. In this condition, single-stranded linear DNA, number of nucleotides minus 1. And in this condition, it would be equal to number of nucleotides. But be very careful that what is given in the question. They have given the number of base pairs or they have given the number of nucleotides or bases. Suppose, for instance, there is a question which says that there is a double-stranded linear DNA and that double-stranded linear DNA has one, you can say, chalo, let's take 100 base pairs. 100 base pairs. So, you'll have to convert these base pairs first into nucleotides. Please don't do that. You directly say 100 minus 2 comes out to be 98. No. Base pairs needs to be first converted into nucleotides. So, one more thing, that if we are talking about the double-stranded condition, then the nucleotides would be present in the pairs. That means in the double-stranded condition, we would be having base pairs. And in the single-stranded condition, we would be having only nucleotides or bases, no base pairs. So here it is given that there are 100 base pairs. So first, convert it into number of nucleotides. That is 100 into 2, because one pair contains 2 nucleotides. So 100 base pairs will be having 200 nucleotides, or you can say 200 bases. 200 nucleotides or 200 bases, not base pairs. And now we can calculate the number of phosphodiester bonds. Number of phosphodiester bonds in this case will be equal to number of phosphodiester bonds will be equal to 200 minus 2. That comes out to be 198. Okay, so this is how you can directly apply these formulas to calculate the number of phosphodiester bonds. And what will be the number of glycosidic linkages? Number of glycosidic linkages will be equal to number of nucleotides. Always, because one nucleotide contains one glycosidic linkage. But phosphodiester bond is present between two nucleotides. So number of n glycosidic linkages will always be equal to number of the nucleotides. And be careful, you have to first convert the base pairs into the nucleotides. And if the question is directly saying that nucleotides, then there is no need for conversion. Okay, let's move further. Yes, This is the important data in NCERT, beta. We can calculate the length of DNA either in the form of base pairs if it is a double-stranded condition, or in the form of nucleotides or bases if there is single-stranded condition. So let's learn these data along with me because they are important. First organism given here is phi 174. And phi 174 is a bacteriophage. That means it is a virus. And what sort of DNA it has? It has single-stranded circular DNA. That means this type. That means it would not be having base pairs. It would be having only nucleotides or bases. And the number of nucleotides are 5386 nucleotides or bases. Repeat with me. 5386 nucleotides, not base pairs. Then the second organism given here is lambda-phage. This is also a bacteriophage or virus which has double-stranded linear DNA. That means this type. And it has 48502. Learn with me. 48502 base pairs. Base pairs, not nucleotides or bases. Then E. coli. E. coli is a bacteria having double-stranded circular DNA. Double-stranded circular DNA, that means this kind of condition. And it has 4.6 into 10 raised to the power 6 base pairs. And the last is human genome. What is the meaning of genome? Genome represents the haploid content. Genome represents the haploid content. That means the number of chromosomes in our sperm or ovum are 23 and the DNA content present in it has this condition. That is 3.3 into 10 raised to the power 9 base pairs. Okay, 3.3 into 10 raised to the power 9. If it is asked that what are the number of base pairs in a human diploid cell, then in a human diploid cell or any mammalian diploid cell, the number of base pairs would be 2 into 3.3 into 10 raised to the power 9. That comes out to be 6.6 into 10 raised to the power 9 base pairs. Okay, so in a human diploid cell, the number of base pairs would be 6.6 into 10 raised to the power 9, and in the haploid cell, it would be 3.3 into 10 raised to the power 9. And you can carefully read the question whether the number of base pairs are asked or the number of nucleotides are asked. If the question says that what is the number of nucleotides in a sperm of humans, so humans have 3.3 into 10 raised to the power 9 base pairs. So the number of nucleotides in the sperm would be 6.6 into 10 raised to the power 9. But if the question is saying that calculate the number of base pairs in the diploid cell, that would be 6.6 into 10 raised to the power 9. And what would be the number of nucleotides in the human diploid cell? 2 into 6.6 into 10 raised to the power 9. Okay? So read the question carefully.
पापा फीस ज्यादा है
बेटा
याद है बोनस के कुछ पैसे
So now we know what is the structure of DNA.